The next presentation, Dr. Juhi. Role of human corneal MiRNA 184 in fungal keratitis.
Good afternoon. I'll be presenting the role of human corneal MiRNA 184 in fungal keratitis. So as we know, corneal blindness is one of the leading causes of blindness, second to cataract in developing countries. In corneal blindness, we see keratitis, and in keratitis also in developing countries, we see fungal keratitis being the common one. MicroRNAs, what are microRNAs? They are a class of small single-stranded non-coding RNA molecule. It regulates the gene expression at post-transcription level by binding to a target miRNA. So in those miRNAs, we are studying miRNA 184. 184 plays a crucial regulatory role in several ocular diseases. Its mutation is seen in keratoconus. However, the role of miRNA 184 in fungal keratitis has not been studied. So in our study, we aim to identify differential expression of miRNA 184 to identify the targets of miRNA by qPCR and to see its clinical utility. For sample collections, we collected corneal swabs as well as corneal tissues. Corneal swabs were collected at the first visit when patients came smear positive for fungus, and after starting the therapy, first follow-up corneal swabs were collected. For tissues, we collected the post-transplant corneal buttons which were positive for fungus. As controls, we used the human donor corneas acquired from an eye bank. In these samples and controls, we did RNA isolation and miRNA sequencing. For swabs, the samples were frozen in zymobuffer, thawed, they were transferred, trizol was added, they were centrifuged, and the swabs were rubbed to the wall of the tube to release the cells, and RNA was isolated. For tissues, again a trizol-based manual method was done. Samples were stored in trizol, thawed, chloroform was added, aqueous phase was separated by centrifugation, RNA was quantified using nanodrop. For target prediction, we used an online tool, it's a MiRNA based tool. For functional analysis, a gene pathway network was constructed using a cytoscape. These are all online tools. Then real-time qPCR was done using the kits that are available, and the expression level of miRNA was compared with an internal control. U6 is an internal control. These were the equipments. So by Mann-Whitney U-test statistical analysis was done, and p-value of less than 0.05 was considered significant. So what we saw in our results was the differential expression analysis showed that miRNA 184 was highly down-regulated when the infection presented at first presentation and in the poor outcome samples, whereas it was up-regulated in controls and the patients who were responding well to the treatment. Another thing we noted, when we saw the target predictors, we saw two pathways, P13 AKT signaling pathway and JAK-STAT pathways, which were our top hit pathways, which were the targets for miRNA 184. So coming to the discussion, we know fungal keratitis an infective disease. It rapidly progresses. It can cause corneal opacity and loss of vision. Several miRNA with profound changes in expression were found in infected cornea, suggesting that they do have a regulatory process in its pathogenesis. Expression of miRNA 184 is abundant in normal cornea, was dysregulated in keratitis, since they may be involved in corneal epithelial cell proliferation, migration during wound repair. Another study which is done on mice in 2006, they saw that miRNA 184 was 89-fold higher in the epithelium of a mice, and in contrast, its expression was absent in limbal conjunctival epithelium. In our study, the differential expression analysis, we identified that miRNA 184 is highly up-regulated in control samples but down-regulated in poor outcome ones. JAK-STAT and P13 pathways were a significant one. So we conclude that it has a significant role in these two pathways, which will provide a therapeutic value in future. Like in cancer studies, for example, they are doing a study in breast cancer in which MiRNA 10B anti-MiRNA 10B nanoparticles they are it is under process, which can have a therapeutic role. So we can have a targeted therapy against these miRNAs. And other than that, it has a prognostic value wherein if we can decide if it is down regulated, we can undergo surgical treatment earlier than to wait for it to get even worse. So the limitation of my study is it's done on a small sample size, so further extensive validation is required. These are my references. Thank you.
Thank you. Any? See this MiRNA 184.
Yes, sir.
There are previous studies which have already shown that it is downregulated in any sort of inflammation in the eye. So taking up fungal, if you had taken up bacterial, you would have got the same downregulation. If you had taken up any other inflammatory, you would have got the same downregulation. So I don't think your study concludes that targeting this as an approach. Targeting this would be an approach for any inflammatory corneal condition.
Yes, sir. But we do also have a prognostic value to it. So if at the first follow-up we see it's still down-regulated, maybe we not wait for it to perforate or just try more medical treatment, or maybe we can intervene at an earlier stage so that...
Yeah, actually, see, this is the disconnect between lab and clinical, because in a fungal, you're seeing a deterioration clinically, even without a slit lamp or anything, you can make out a deterioration. You don't wait for an miRNA 184 to tell you to be more aggressive or whatever. You go by the clinical picture. So it may not have too much of a clinical significance, though it does have a, what you're proving is correct in the lab. Thank you.
Thank you.